If I scrape my knee or I burn the roof of my mouth on hot coffee, the tissue damage is just temporary.
The body just fixes it.
Right. The surrounding epithelial cells, they recognize the deficit, they enter a phase of the cell cycle called mitosis, they duplicate their DNA, and they divide.
The gap gets filled.
Exactly. The tissue regenerates. But the brain operates under an entirely different set of rules, doesn't it?
It really does. The vast majority of neurons in the human brain are what we call postmitotic.
Postmitotic?
Yeah. Once they reach maturity during our early developmental stages, they exit the cell cycle permanently. They do not divide.
Which sounds like a terrible design flaw at first glance.
Well, the evolutionary logic behind this is actually quite beautiful, even if it leaves us incredibly vulnerable.
How so?
The brain functions as this hyper-complex network of electrical connections, synapses, and established pathways. And those encode our memories, our learned behaviors, and our fundamental personalities.
Right, the wiring makes us who we are.
Exactly. If neurons were constantly dividing and moving around like skin cells, the physical architecture of those memories would just be constantly disrupted. Stability is the absolute prerequisite for consciousness and memory.
Welcome to the Deep Dive. I'm Satosha.
And I'm Satoshi.
We are dropping you straight into a reality that sounds like, a Hollywood spy thriller, but it's actually unfolding right now at a microscopic level inside the human body.
It really is.
We are diving into a recent paper published in the journal Nature. It's by a collaborative team from UC Berkeley and UT Southwestern, and the narrative here is just stunning.
It's incredible.
We're going to explore how a quest to cure neurodegeneration accidentally uncovered this built-in self-destruct mechanism for cancer.
The stakes of this research, they really cannot be overstated. When a paper lands in Nature, we are not looking at some, incremental update to a niche subfield.
Right, it's a huge deal.
Nature is the absolute apex of scientific publishing. For context, the rejection rate there hovers around 92%.
Wow, 92.
Yeah. So a publication in this journal means the data has survived an absolutely brutal gauntlet of peer review. It indicates that the findings fundamentally shift our baseline understanding of biological reality.
That's wild.
The Berkeley and UT Southwestern researchers, they've mapped a previously invisible mechanism that actually governs life and death at the cellular level.
Man, the biological trade-off is just staggering when you frame it that way. We essentially trade the ability to heal for the ability to remember.
That's a great way to put it. We gain a stable identity, but we lose the capacity for cellular replacement.
When that neuron begins to fail, there's literally no backup system ready to take its place.
None. Which brings us to the devastating reality of neurodegenerative diseases. They are all characterized by the progressive, catastrophic loss of these irreplaceable cells.
That's heartbreaking.
It is. Because when a patient loses these neurons, they are not just experiencing physical degradation. They are losing the structural foundations of their identity.
Yeah, the loss of motor control, the erosion of memory, the inability to recognize loved ones.
Exactly. Those are the direct macroscopic results of microscopic cells dying and leaving a permanent void in the neural network.
Knowing that the stakes are that high, the preservation of human identity itself, it makes total sense that billions of dollars have been poured into stopping neurodegeneration.
Oh, absolutely.
But the prevailing scientific strategy for decades was, it was focused heavily on this concept of cellular waste management. Let's examine that old paradigm for a second.
The garbage theory.
Right, the garbage theory. For years, the narrative was that neurons were dying because microscopic garbage was just piling up inside them.
Yeah, that theory has dominated the field. As any cell performs its daily metabolic functions, it generates waste.
Like exhaust from an engine.
Pretty much. It produces misfolded proteins, oxidized lipids, and damaged organelles. Now, in a healthy scenario, the cell breaks this waste down and recycles it.
But in a diseased brain, that doesn't happen.
No. Pathologists constantly observe massive accumulations of this biological trash. In Alzheimer's, we see amyloid beta plaques and tau tangles.
And in Parkinson's, it's Lewy bodies.
Exactly. Lewy bodies composed of a protein called alpha-synuclein. So the logical deduction for decades was that this accumulating waste was toxic, and its mere presence was choking the neuron to death.
Which makes sense on the surface. If the underlying assumption is that the garbage itself is the murder weapon, then the therapeutic strategy becomes obvious.
We build a better broom.
Exactly. You try to build a better microscopic broom, you develop drugs that bind to the amyloid plaque to dissolve them, or you try to stimulate the cell to just flush the tangles out.
What's fascinating here is that the Berkeley and UT Southwestern researchers, they took a huge step back and challenged that core assumption.
They totally inverted the question.
They questioned the causality entirely. Instead of asking how to clear the accumulating waste, they asked, well, what is causing the waste management system to fail in the first place?
Right. The presence of garbage is merely a symptom.
Yes. The true pathology lies in the failure of the incinerator itself. And to understand their breakthrough, we really have to zoom in way past the brain tissue into the specific cellular organelle responsible for waste disposal.
The lysosome. For anyone listening who hasn't looked at a diagram of a cell since high school biology, let's build a clear mechanical picture of what a lysosome actually is.
It's vital to visualize this.
Because the cytoplasm, the fluid inside the cell, it is not just an empty swimming pool.
Not at all.
It is this dense, highly structured, incredibly crowded environment filled with various micromachines. And floating within that crowded space are lysosomes.
The repair mechanism.
Right. If I am visualizing the inside of a single cell in my body right now, what are the actual biophysics of this reactor?
It comes down to basic chemistry, specifically pH and containment. So the main fluid of your cell, the cytoplasm, where all your DNA and machinery just float around, that sits at a neutral, comfortable pH of about 7.2.
Okay, 7.2. Pretty neutral.
Right. But inside the thin boundary of the lysosomal membrane, the pH is maintained at roughly 4.5. It is incredibly acidic.
And pH is logarithmic, so a drop from 7.2 to 4.5 isn't just, a little bit more acidic. It is orders of magnitude more corrosive.
Yes. It is an environment expressly designed to strip chemical bonds apart. And to maintain that extreme gradient against the natural flow of nature, the cell uses these tiny biological motors called vacuolar AT-passes.
Vacuolar AT-passes. What do those do?
They're literally physical rotors embedded in the membrane. They constantly burn energy to pump protons from the neutral cytoplasm into the lysosome, artificially driving the acidity up.
So you literally have a microscopic balloon filled with acid.
Filled with acid and armed with over 60 different hydrolytic enzymes.
Wow, 60.
Yeah, and these are molecular shredders specifically folded to operate strictly in high-acid environments. Their entire biological purpose is to break down proteins, lipids, and carbohydrates.
Which poses an obvious, glaring engineering paradox.
Right.
Because the hydrolytic enzymes inside the lysosome are designed to shred proteins and lipids. But the protective membrane holding those enzymes
is made of proteins and lipids.
Exactly. It's like building a vat to hold acid, but you construct the vat out of the very metal the acid is designed to melt.
That is the exact biophysical constraint we're talking about. The limiting membrane of the lysosome is perpetually under threat from its own contents.
It's fighting itself.
Constantly. And the stress isn't just internal either. The lysosome is tasked with breaking down toxic aggregated materials from the rest of the cell. Sharp, misfolded protein crystals are constantly being shoved into it.
So it's taking physical damage, too.
Absolutely. Microlesions, nanoscale ruptures, and physical stretching happen constitutively just from a cell doing the normal work of staying alive.
But there's another layer to this that makes the architecture even more confusing. The cell doesn't just isolate this dangerous reactor in a corner somewhere. It actually uses it as a central command post.
Yeah, this is what makes containment so critical. The surface of the lysosome is an essential signaling hub. It serves as the physical activation platform for a complex called mTORC1.
mTORC1. I've heard of that in longevity research.
Yes, it's the master kinase, the central switch that drives all cellular growth, metabolism, and energy distribution.
And it sits right on the surface of the bomb.
It sits right on the surface. So the cell is entirely dependent on this organelle for survival and growth, but the organelle itself is a perpetual liability.
This brings us back to the water balloon analogy. Think about the cells in your eye right now registering the light in the room. Inside those cells, these highly acidic reactors are perpetually taking microdamage. So how does the cell not just dissolve itself every few hours?
Right.
Because if that containment fails and those highly acidic enzymes leak out,
system failure is rapid and catastrophic. If a lysosome ruptures completely, the pH of the entire cell begins to drop. Those hydrolytic enzymes escape into the cytoplasm and start digesting the cell's essential machinery from the inside out.
it eats itself.
Literally. And when this happens, the cell triggers what we call necrotic or apoptotic cell death. It recognizes that containment has failed and essentially executes a self-destruct sequence to prevent the toxic acidic spill from spreading to neighboring tissue.
So logically, there has to be an automated, highly sensitive repair protocol, something that detects the micro tears and deploys a physical sealant before the whole system goes critical.
Right. And biologists have known for a long time that such a system had to exist theoretically. If it didn't, complex life couldn't survive past a few cell divisions.
It just wouldn't work.
But for decades, how the cell actually detected the breach and managed to deploy a microscopic patch in a matter of seconds was a complete black box.
And to understand how a complex system works, sometimes you have to intentionally break it.
Exactly.
The methodology the researchers used to look inside this black box is fascinating because of how brute force it is. They didn't just passively observe cells, they executed a CRISPR interference screen.
Yeah, and to contextualize why they needed such an aggressive approach, we have to look at what was already known in the field. We knew about a set of cellular machinery called ESCRT.
ESCRT.
You can think of ESCRT as the cell's premier topological remodeling apparatus. We knew it had the physical capacity to bend, cut, and patch lipid membranes.
That's the cellular sealant.
It is the physical patch itself. We had mapped its role in normal programmed events like when a cell divides into two, ESCRT comes in to pinch the membrane shut, or when a virus like HIV hijacks the cell, it uses ESCRT to bud out of the membrane.
But those are scheduled events. I mean, a programmed cell division is a massive, highly coordinated, slow-moving logistical effort. The cell knows exactly where and when it needs the ESCRT sealant.
Right, but emergency repair is entirely different. It is stochastic. It happens randomly, unpredictably at microscopic scales. For a sudden lysosome rupture, you need an immediate, localized, massive amplification of signals out of nowhere.
The alarm has to be instant.
Exactly. We knew ESCRT was the patch, but we had absolutely no idea what the alarm transducer was. What was the missing link that screamed for ESCRT to arrive at a tiny microlesion in a fraction of a second?
So to find that alarm, the researchers built a highly sensitized genetic screen. They took a human leukemia cell line called K562, but they didn't just use standard cells, they engineered them. They permanently removed a specific cholesterol transporter called MPC1. Why start by breaking a cholesterol pump?
By removing that single NPC1 transporter, they forced the cells to accumulate an abnormal, rigid lipid profile, specifically in their lysosomes. They deliberately made the lysosomal membranes biophysically fragile.
Oh.
It stripped away the cell's natural chemical buffering and safety margins. They created an environment where the lysosomes were already stretched thin, teetering right on the brink of structural collapse.
They basically built a house out of dry tinder, and then they introduced a chemical stressor called LMO to strike the match.
LMO is a very elegant chemical tool. When you introduce it to a cell culture, it freely diffuses across the outer cell membrane and floats through the neutral cytoplasm, doing absolutely nothing. It is completely inert.
But when it crosses into the lysosome,
when it hits that extreme pH 4.5 environment, the acid triggers a rapid condensation reaction. The LMO molecules chemically link together, forming rigid, highly structured polyleucine oligomers.
it's like microscopic ice crystals suddenly freezing and expanding inside a closed glass bottle.
And just like ice in a glass bottle, the physical expansion of these oligomers aggressively punctures the fragile lysosomal membrane from the inside out. The researchers used loam to simulate acute, progressive organelle injury across millions of cells simultaneously.
That's wild. They have millions of fragile cells, and they are intentionally popping the lysosomes from the inside out. And this is where the ultimate irony of modern biology comes in. To figure out how the physical membrane heals, they deployed a tool called DCAS9 CRAB.
Right. This is a modified, non-cutting form of CRISPR, usually referred to as CRISPR interference or CRISPR.
Because standard CRISPR is famously a genetic editor. It cuts DNA, but this version doesn't cut.
No. It acts as a highly targeted silencer. Instead of severing the genetic code, DCAS9 CRAB binds to a specific gene and physically blocks the machinery that tries to read it. It is literally a volume knob turned to zero.
Okay, so they turn the genes off.
Right. The researchers took a massive library of guide RNAs and used this system to methodically turn off every single gene in the human genome, one by one, across different populations of these stressed K562 cells.
So just think about the architecture of this experiment for a second. They are using CRISPR, the ultimate informational software editor, not to cure a genetic disease, but as a brute force stress test switch.
Yeah.
They are using a software blackout to discover a physical hardware maintenance mechanism.
It is an ingenious inversion of how we normally think about genetics. They subjected these varied populations of cells to alternating pulses of the alum stressor over 8 days. Then they simply looked at the survivors. They were hunting for sensitizing genes.
Meaning, if they turned off gene X, and that specific batch of cells immediately died when exposed to the lysosome stressor, then gene X must be a critical component of the emergency repair alarm.
Exactly. And the output of the screen was a massive, high-dimensional data set. Millions of data points mapping survival against genetic silencing. To make sense of all that noise, they fed the data into bioinformatic clustering models, specifically the Louvain algorithm, to look for hidden associative networks.
And then out of that massive mathematical abstraction, a highly significant, completely novel central node emerged, a protein called TFG.
And to say TFG was a surprise is an understatement. Prior to this data, TFG was considered a very mundane, localized structural protein.
What was it known for?
Its only known job was managing routine, low-stakes vesicle traffic over at the endoplasmic reticulum. No one in the structural biology community suspected it was the central orchestrator of emergency organelle repair.
But the bioinformatics don't lie. Without TFG, the lysosomes ruptured, the ESCRT sealant was never deployed, and the cells died rapidly. So now we have the actor, we have the stage. This allows us to watch a real-time hardware repair protocol execute in milliseconds.
Yeah.
The researchers named this the laser protocol. And instead of breaking this down into rigid textbook steps, let's just follow the physical cascade as it happens in real time.
Okay, so the sequence initiates the precise moment the membrane is compromised. When the LMA crystal, or in a natural environment, a toxic misfolded protein punctures the lipid bilayer, there is a sudden, localized environmental shift.
Because the inside is different from the outside.
Right. The inside of the lysosome is packed with calcium. The physical breach allows a sudden microplume of calcium to spill out into the cytoplasm.
This tiny chemical spill is the absolute initiating trigger. It's the smoke detector going off.
And the researchers proved this causality beautifully. They pre-treated a batch of cells with a chemical chelator called BAPTA-AM.
BAPTA-AM.
Yeah. You can think of a chelator as a highly specific chemical sponge. BAPTA-AM instantly binds to and neutralizes any free calcium the exact moment it appears.
So the membrane still breaks, but the calcium is instantly soaked up before it can travel.
Exactly. And when they did that, the entire repair sequence stopped dead. Nothing assembled. The cell just died. Conversely, they found that if they artificially forced a calcium release using a specialized drug, without actually breaking the membrane at all, the repair machinery still rushed to the site and began to assemble.
Wow. The calcium plume is the definitive signal. So the alarm goes off. What happens in the immediate microsecond after the spill?
A dual action response occurs almost simultaneously. First, the endoplasmic reticulum, the ER, which is a massive, sprawling network of folded membranes that snakes throughout the cell, physically moves its architecture toward the site of the damage.
Wait, it actually shifts its physical structure?
Yes, it creates a localized contact site. It tethers itself directly to the broken lysosome using specialized bridging proteins called VAPs.
It's like an emergency response command center pulling right up alongside a damaged nuclear reactor to establish a supply line.
A very apt metaphor. And at the exact same time the ER is pulling up, a secondary signaling pathway called CSM activates on the surface of the broken lysosome.
What's that do?
Specialized marker proteins belonging to the ATG8 family, specifically molecules named LC3B and GABARAP, are covalently lipidated directly onto the damaged surface.
Hold on, what does covalently lipidated actually mean in this context?
It means they are not just loosely floating nearby. The cell is physically gluing these ATG8 marker proteins directly into the structural lipid molecules of the broken membrane. It is literally painting a bright biochemical target directly over the microscopic hole.
Okay, so the command center is tethered nearby, and the exact site of the breach is painted with permanent markers. This is where our unexpected hero, the TFG protein, comes into play, because TFG normally lives on the ER.
Right. Its day job is on the endoplasmic reticulum. But because the ER has now physically tethered to the damaged lysosome via those VAP proteins, TFG is brought into extreme spatial proximity to the crisis.
It's right next door now.
Exactly. It senses the painted target, leaves its normal post, and moves across the tiny gap to the damaged surface.
And it anchors itself there. It docks using a highly specific part of its physical structure called the LIR2 motif. It reaches out and binds directly to those ATG8 proteins that were just glued over the hole.
Yes.
But this is where the biophysics gets deeply confusing. I was reading the supplementary data in the research, and I actually thought it might be a typo.
Yeah.
The mathematical strength of this bond between the TFG responder and the damaged surface is absurdly weak.
Oh, it is thermodynamically unstable. The binding affinity, the dissociation constant, sits in the 60 to 100 micromolar range.
To ground that for you listening, normal, permanent structural bonds in a cell, like the ones used by classic autophagy receptors to hold things together, are usually around 1 or 2 micromolar.
Right.
This emergency bond is 30 to 50 times weaker. It's like trying to patch a high-pressure water main with a wet Post-it note. Why would evolution select for the weakest possible grip during a catastrophic emergency?
It seems entirely counterintuitive until you look at it through the lens of biological noise calibration. If that initial LIR2 bond were incredibly strong, like a permanent molecular superglue TFG, would become a massive liability.
How so?
It would constantly stick to everything in the cell that had even a trace of baseline ATG8 on it.
You'd have constant misfires.
Exactly. You would have emergency cascades triggering during routine metabolic fluctuations. The repair machinery would assemble when there was no actual breach, wasting massive amounts of cellular energy and potentially gumming up normal, healthy organelle function.
So the extreme weakness is actually a safety mechanism. It's calibrated specifically for a transient, easily reversible interaction. It needs to easily attach, but also instantly fall off if the calcium alarm turns out to be a false positive.
It is the biological equivalent of a provisional hold. But you're absolutely right about the physics.
Yeah.
A single weak bond cannot possibly hold a massive multi-protein repair complex together during a violent membrane rupture.
Which is why TFG doesn't act alone. It utilizes a principle called avidity. And this is perhaps the most elegant geometric trick in the entire protocol.
It's beautiful, really. Once a single TFG molecule is loosely anchored to the surface, it utilizes two other regions of its own structure, the PB1 domain and the coiled coil domain. You can visualize these domains as highly specific interlocking puzzle pieces.
So you have multiple TFG molecules loosely bouncing around the painted target, attaching and detaching.
But when a few of them happen to anchor near each other in the exact same orientation, those coiled coil domains snap together, they multimerize, they physically interlock to form a highly ordered octameric ring, an 8-part circular structure composed entirely of TFG molecules.
This is the power of avidity. A single weak bond is useless. But when you lock 8 of them together into a rigid geometric ring, you suddenly create massive structural stability. I picture it like Velcro.
Oh, that's a good analogy.
Right. One tiny plastic hook on a strip of Velcro is incredibly weak. A light breeze could detach it. But when you have thousands of those tiny weak hooks arranged together in a dense geometric array, they can support the weight of a person.
Avidity is the transformation of many weak, transient interactions into a single, unbreakable macrostructure. By oligomerizing into this rigid octameric ring, the TFG complex firmly and securely anchors itself directly over the nanoscale lesion, entirely resisting the fluid turbulence of the leaking membrane.
It sets up an immovable perimeter around the damage. And that rigid structure is what finally allows the cell to deploy the actual sealant.
Right, because the entire purpose of forming this massive TFG ring is payload delivery. It acts as a highly stable landing pad. The outer edge of the TFG ring presents a dense, outward-facing array of specific amino acid sequences called PSAP motifs.
These SAP motifs, and these are perfectly shaped to bind to a floating protein called TSG101.
Correct. TSG101 is the essential initiator for the ESCRT machinery. Under normal conditions, TSG101 is just floating in the cytoplasm, ignoring the weak background noise of the cell.
Just drifting.
But when it floats past a damaged lysosome and encounters this dense, highly stable, blindingly obvious array of PSAP motifs presented by the TFG ring, it binds securely.
The landing pad catches the initiator.
And once TSG101 is securely anchored, the kinetic threshold is crossed. The cascade becomes unstoppable. TSG101 calls in the rest of the ESCRT-I complex, which sequentially recruits ESCRE3.
And ESC3 is the actual physical patch. We are finally at the point of repair.
Yes. ESCRT subunits arrive at the site and begin to polymerize. They linked together, forming flat, spiraling filaments directly on top of the broken lipid bilayer.
Spiraling filaments.
Think of a microscopic coil spring lying flat against a surface, slowly tightening. As the ESCRT spiral grows and tightens, it physically draws the broken, ragged edges of the lipid membrane together.
It pulls the biological fabric closed.
It seals the nanoscale hole. The leakage of acid and calcium instantly stops. The internal pH stabilizes. The lysosome is saved from catastrophic rupture, and the cell survives.
And this entire massive architectural undertaking, the calcium alarm, the ER tethering, the target painting, the weak docking, the geometric ring formation, the signal amplification, and the spiraling filament patch happens in milliseconds to minutes.
It's remarkably fast.
The precision is just staggering. Think about how perfectly the cell filters out all the chaotic background noise of normal metabolism to focus entirely on this localized damage and execute a multi-stage physical repair. The researchers realized that this internal tearing, this constant microscopic leaking, was the true root cause of the accumulating garbage.
And the eventual death of the neuron, yes.
The theory makes perfect logical sense. If the incinerator is broken and leaking acid, the trash is obviously going to pile up and the factory will eventually burn down.
Exactly.
But testing that theory, that presents an astronomical logistical challenge. We are talking about detecting a microscopic rip in the lipid bilayer of an organelle that is already just a fraction of the size of a single cell.
It's mind-boggling.
How do you even architect an experiment to observe that in real time?
Observing this phenomenon requires pushing the absolute boundaries of modern microscopy and high-dimensional data analysis.
Because you can't just look through a regular lens.
No, you cannot simply place a neuron under a standard light microscope and wait to see a tear. The events occur on a nanometer scale and unfold in a matter of milliseconds.
So what did they do?
The researchers had to utilize advanced techniques like live cell fluorescence, confocal microscopy.
Meaning they made it glow?
Yes. They essentially engineer the cells so that specific proteins involved in the lysosome membrane glow under certain wavelengths of laser light.
So they are tagging the structural components of the organelle with biological highlighters.
Precisely. They introduce fluorescent markers that specifically bind to the acidic interior of the lysosome. If the membrane tears, the pH changes instantly, and the fluorescence just vanishes.
Oh, wow. So it acts as a visual alarm bell.
It does, but capturing that visual data is only a fraction of the challenge. The sheer volume of chemical noise inside a living cell is overwhelming.
Just too much going on at once.
There are millions of interacting molecules, thermal fluctuations, and overlapping signaling pathways occurring every single millisecond. Isolating the specific biochemical cascade that happens the exact moment a lysosome tears requires filtering out a massive amount of background noise. And returning to the experiment at hand, the researchers from Berkeley and UT Southwestern utilized their advanced data filtering to look past all that cellular noise.
Right, back to the microscopic leaking incinerator.
They successfully isolated the signal of the tearing lysosome, but they didn't just observe the tear and watch the cell die. They observed a miraculous countermeasure.
They saw the cell fight back.
They witnessed the cell actively and aggressively fighting back against the breach.
It's just mind-bending in its complexity.
It really is a marvel of biology.
But the most crucial variable here is time. I want to ask for some clarification here, because I think it is important to draw a hard line between this type of emergency cellular repair and the kind of genetic manipulation that usually dominates the headlines, like CRISPR.
Right. People confuse them.
Because are we editing DNA here? They operate on entirely different temporal dimensions, don't they?
The distinction is absolutely vital. CRISPR-Cas9 is a revolutionary tool, yes, but it is fundamentally a mechanism for editing the genetic code found within the nucleus of the cell.
It's rewriting the software.
If we use a structural analogy, using CRISPR is like discovering a flaw in a city's infrastructure, realizing you need to rewrite the architectural blueprints at City Hall, getting the new blueprints approved, and then waiting for new manufacturing plants to build the replacement parts.
Rewriting the blueprint of a cell takes hours, if not days, right? Because you have to wait for the DNA to be transcribed into RNA, and then for that RNA to be translated into brand new proteins by the ribosome.
Exactly. The central dogma of molecular biology, DNA to RNA to protein, is a slow, deliberate process. If your lysosome has just ruptured and highly acidic, destructive enzymes are actively dissolving the internal architecture of your irreplaceable neuron.
You don't have time for all that.
You do not have hours to wait for City Hall to draft a new blueprint. The neuron will undergo apoptosis in a matter of minutes.
The response has to be instantaneous.
Which is why the laser, TFG, and ESCRT pathway is so incredibly critical. It does not rely on genetic transcription. It relies on proteins that are already manufactured, constantly patrolling the cytoplasm, fully primed for activation.
It's already on duty.
When the tear occurs, the conformational changes and the phase separation happen in milliseconds. The physical patching of the membrane happens in seconds. It is a reactive biophysical emergency response that prioritizes immediate, desperate survival over long-term adaptation.
The researchers mapped this entire millisecond by millisecond emergency sequence. They finally understood exactly how a healthy cell manages to survive these microtears without dying.
They solved a huge part of the puzzle.
And the logical next step in their original mission, curing neurodegeneration, would be figuring out how to enhance this system.
Right.
Like if we could develop a drug that makes the laser beacon more sensitive or helps TFG condense faster, maybe we can help failing neurons patch their leaks before the toxic garbage piles up.
But, scientific discovery is rarely a linear journey from hypothesis to therapeutic application.
It's chaotic.
It is highly chaotic. When you map a fundamental mechanism of cellular survival with this much precision, the implications inevitably bleed over into totally unexpected territories.
Which brings us to the serendipitous pivot of this entire narrative.
Yeah.
And I just marvel at the serendipity here.
It's amazing.
They were looking at neurodegeneration, but the data they were analyzing regarding how cells prevent their own destruction pointed a glaring arrow toward an entirely different field of medicine, oncology.
Yes.
The researchers realized that this repair mechanism isn't just relevant to neurons that we desperately want to save. It is highly relevant to cells that we desperately want to destroy.
And that realization is just so powerful. It starts as a chaotic, high-dimensional field of signals competing models existing simultaneously without structure.
Honestly, it connects directly to how these deep dives are created for you. Every deep dive begins just like this biological research. A chaotic field of papers, philosophy, and technical references.
That's a massive puzzle.
The process is all about discovering hidden associative threads and recursively refining them into coherence. AI systems help explore massive conceptual spaces and surface connections, but the raw generation remains noisy.
You have to find the signal in the noise.
The real challenge is recursively constraining and refining ideas into clarity through rewriting, compression, debate, verification, and just obsessive iteration. Sometimes a single metaphor is refined repeatedly until it clicks.
It takes a lot of work to make it clear.
We then use visual storytelling tools like Mosaic to create minimalist motion graphics and restrained typography that respect silence, pacing, and cognitive clarity. It's all about transforming informational chaos into a precise line of insight.
And that precise line of insight, in this case, completely changed their focus.
So let's return to that key insight. How does a brain cell's emergency repair system connect to cancer?
To comprehend the logic of this pivot, we must look at the fundamental metabolic nature of a cancer cell.
Okay, let's look at it.
A cancer cell is defined by a complete disregard for the regulatory signals that normally govern the cell cycle. It divides uncontrollably, it replicates its DNA with chaotic speed, and it is in a state of constant hyper-accelerated metabolic overdrive.
The cellular machinery is basically running at maximum capacity with the safety limiters completely removed.
Exactly. This phenomenon, which is often related to the Warburg effect, means the cancer cell is consuming massive amounts of glucose and synthesizing proteins at a staggering rate to fuel its rapid division.
But I imagine this hyperacceleration comes with a massive cost.
It does. The cancer cell is incredibly messy. It generates an unprecedented volume of misfolded proteins, damaged organelles, and toxic metabolic byproducts.
So the cancer cell is producing a catastrophic amount of biological garbage, which means its waste management system, its lysosomes, must be working absolute overtime.
The stress placed on the lysosomal network of a cancer cell is just immense. The incinerators are packed beyond capacity, and the internal environment becomes hyperacidic to compensate for the massive influx of waste.
That sounds unsustainable.
Because of this extreme, constant physiological stress, the lipid bilayers of a cancer cell's lysosomes are highly unstable. They're experiencing constant, severe microtears.
Wait, if the incinerators in a cancer cell are constantly rupturing and leaking corrosive acid into the cytoplasm, why doesn't the cancer cell just digest itself and die?
That exact question was the profound aha moment for the Berkeley and UT Southwestern team.
Really?
The cancer cell does not die because it is utterly and completely dependent on the ASER, TFG, and ESCRT repair pathway.
Oh, wow.
Because its lysosomes are constantly tearing, the cancer cell's emergency response system is operating at maximum output 24 hours a day, patching thousands of microtears every minute just to prevent catastrophic autolysis.
The realization here is staggering. This elegant, rapid response repair system, which evolved to protect our most precious, irreplaceable neurons from isolated accidents, has basically been hijacked.
Yes, it has.
The tumor is using our own cellular fire department as its ultimate shield against its own chaotic metabolism.
The cancer cell's greatest metabolic weakness, its massive production of toxic waste and fragile lysosomes, is completely masked by its reliance on the ESCRT machinery. The repair pathway is the only thing preventing the tumor from collapsing under the weight of its own toxicity.
Here's where it gets really interesting. This insight completely upends the traditional paradigm of oncology, doesn't it?
It absolutely shifts the entire landscape.
Up until now, prevailing cancer research strategies were mostly about identifying cancer cells and throwing external poison at them.
Right, chemotherapy and radiation.
The foundational logic of chemotherapy is to introduce highly toxic chemical agents into the bloodstream that specifically target rapidly dividing cells.
Traditional chemotherapies like alkylating agents or antimetabolites are essentially blunt instruments. They interfere with DNA replication or cellular division.
Hoping to kill the bad guys before the good guys.
The hope is that because cancer cells divide faster than healthy cells, the poison will kill the tumor before it causes irreparable damage to the surrounding healthy tissue. But the collateral damage is immense.
Yeah, the side effects are brutal.
The toxicity affects hair follicles, the gastrointestinal tract, the immune system, really any system that relies on regular cell turnover.
We are essentially carpet bombing the body and hoping the cancer dies before the patient does. But the implications of this Nature paper suggest a radically different approach.
A targeted self-destruct signal.
We don't need to invent a new external poison to throw at the tumor. We just need to disable the cancer cell's stolen shield.
The therapeutic concept here is breathtakingly elegant. Instead of external poison, this research suggests utilizing specialized small molecule inhibitors.
Drugs precisely engineered to block this system.
Exactly. Drugs designed to bind to and block the function of either the laser complex, the TFG phase separation, or the ESCRT proteins themselves. It is the microscopic equivalent of a targeted sabotage operation.
We chemically jam the biological fire alarm, or basically tie the hands of the emergency responders.
Yes. And when you introduce an ESCRT inhibitor into the biological system, you disable the cellular repair machinery. For a healthy normal cell, this is merely a minor inconvenience.
Because they don't tear that often.
Right. A healthy cell's lysosomes are relatively stable. They tear rarely, so the temporary loss of the emergency repair pathway is manageable. But for the cancer cell, it is an immediate death sentence.
Because the cancer cell is already tearing constantly. If we block the ESCRT proteins, the very next micro-tear in the tumor's lysosome will not be patched.
The tear widens. The highly acidic cocktail of 50 different hydrolase enzymes floods freely into the cancer cell's cytoplasm. The sudden, massive drop in pH and the indiscriminate degradation of the internal structures trigger rapid apoptosis.
It's literally this tape will self-destruct in 5 seconds trope from Mission Impossible, but played out on a microscopic battlefield.
It really is. The cancer cell is forced to execute its own destruction, utilizing the very acid it created to manage its chaotic growth.
It is the ultimate biological judo. We are using the tumor's own aggressive, messy momentum against it. We are not poisoning the cancer. We are simply removing the one specific protein complex that stops the cancer from destroying itself.
The specificity of this approach is what makes it so revolutionary. By manipulating this biological pathway and targeting a vulnerability unique to malignant cells, we potentially bypass all that systemic toxicity associated with traditional chemotherapy.
We are leveraging the fundamental biomechanics of the cell to achieve selective eradication.
Exactly.
Good. To make sure you, the listener, walk away with a crystal clear understanding of this massive paradigm shift, let's do a rapid-fire recap answering the 5 questions for popular media, the 5 Ws.
Sounds good. Let's do it.
Cool.
Researchers at UC Berkeley and UT Southwestern discovered the laser and ESCRT lysosomal repair mechanism.
When?
Just published prior to June 2026.
Where?
In the premium journal, Nature.
And finally, why?
To potentially revolutionize cancer treatment and neurodegeneration by targeting the exact same cellular machinery, literally killing 2 very terrifying birds with one stone.
Let's step back and look at the vast expanse of the biological landscape we have just traversed. We began by looking at the tragedy of neurodegeneration, the slow, heartbreaking loss of memory and identity caused by the inability of neurons to repair their internal structures.
The tragedy of slow decay.
And we arrived at the chaotic, explosive growth of a tumor, desperately patching its own internal structures just to survive its own toxicity. And the connective tissue between these two massive domains of human suffering is a single microscopic mechanism.
Just a few highly specialized proteins rushing to stitch together a leaking lipid bilayer in a matter of milliseconds. If we connect this to the bigger picture, when you view biology through this deeply mechanistic lens, you begin to recognize a profound structural symmetry.
It's beautiful and terrifying all at once.
At the fundamental molecular level, the forces that govern survival and the forces that govern destruction are not opposing armies fighting on a battlefield. They are mirror images utilizing the exact same machinery, the exact same sensors, and the exact same physics.
The biological symmetry is just astounding. The line between preserving a memory and eradicating a tumor isn't a difference in weaponry. It is entirely a matter of signaling.
If we can learn to read the molecular manual accurately enough, if we can understand the precise conformational changes of these proteins, we can intervene with absolute precision.
We can enhance the laser and ESCRT pathway to save the brain, reinforcing the fragile lysosomes of our aging neurons to hold Alzheimer's.
And we can simultaneously deploy inhibitors that block that exact same pathway in malignant tissue, forcing the cancer to drown in its own acidic exhaust.
We are moving past the era of viewing disease as an invading force that must be attacked with external chemicals. We are entering an era of biological engineering, where the cure for our most feared diseases lies not in building new weapons, but in simply understanding the switches of the machines already inside us.
It's a new frontier of medicine.
And that leads to a fascinating, highly provocative implication for you to explore on your own, something that extends beyond both neurodegeneration and oncology. If the stability of the lysosome membrane and the efficiency of the ESCRT repair pathway are the fundamental arbiters of cellular life and death,
Right.
it raises a massive question about the nature of time itself. Could this exact mechanism be the underlying biological clock of aging?
That is a phenomenal avenue for speculation. We know that as the human body ages, we see a systemic increase in cellular senescent cells that have stopped dividing and begin secreting inflammatory signals that degrade surrounding tissue.
The exact trigger for why a cell becomes senescent has been debated for decades.
Consider the physics of the repair we just discussed. The ESCRT proteins pinch the membrane together to seal the tear. But does that repair leave a microscopic scar on the lipid bilayer?
Like scar tissue on skin.
Yes. If a cell experiences thousands of these micro tears over a lifetime, does the lysosome eventually become too rigid, too scarred with patched lipids to function properly?
If the efficiency of the laser and ESCRT pathway naturally degrades over time, or if the accumulation of patched membranes eventually compromises the proton pumps, the slow leakage of acidic enzymes might not be severe enough to trigger sudden cell death.
But it might be the exact trigger that pushes the cell into a state of chronic inflammatory senescence.
It reframes the entire concept of mortality. We don't just get old. We accumulate microscopic scars on the internal incinerators of our cells until the biological fire department can no longer keep up with the structural fatigue.
The implications for longevity research are staggering. If we can pharmacologically tune the sensitivity of the TFG phase separation, could we essentially reset the resilience of the lysosome and delay the fundamental onset of cellular aging?
The potential extrapolations of this single Nature paper touch almost every aspect of human biology. It forces us to reconsider the mechanics of longevity, the origin of inflammation, and the incredibly delicate balance of enzymes and lipids that keep us alive second by second.
It is a profound reminder of the complexity operating beneath the surface of our awareness.
Thank you for joining us on this intense, layered exploration. We unpack the architectural vulnerability of the postmitotic brain, the acidic mechanics of the lysosome, the high-dimensional data filtering required to see a nanometer tear, the biophysics of liquid phase separation, and the metabolic chaos of cancer.
It is a demanding conceptual journey, but understanding these mechanisms is crucial to grasping the future trajectory of medical science.
If you value this kind of uncompromising, rigorous analysis of the forces shaping our reality, we invite you to subscribe to The Deep Dive. You can find us on Spotify, Apple Podcasts, YouTube Music, and value-for-value platforms like Fountain.fm.
And the synthesis does not stop here.
Yep.
We actively encourage you to submit your own research papers, your philosophical frameworks, technical references, obscure readings, and deep conceptual rabbit holes for future deep dives.
The most valuable insights emerge from collaborative synthesis, from taking disparate pieces of high-dimensional information and obsessively refining them until the underlying structure becomes clear. Meaningful ideas emerge through collaborative synthesis between human curiosity, AI acceleration, and recursive refinement. So as you step away from this audio and back into the relentless noise of the modern world, take a moment to consider the silent, invisible miracles happening inside you right now. At this exact second, a microscopic tripwire is detecting a calcium leak, a protein droplet is forming, and a molecular coil is physically suturing a torn membrane just to keep a single memory intact. Keep searching. Keep refining your models of reality. We'll see you on the next Deep Dive. I'm Satosha.
And I'm Satoshi.